go back away when I was a postdoc at Michigan. I tried to persuade him to come and do his grad degree at Michigan with Wes Brown. Sadly for us, and great for him, he elected to come to Berkeley, where he worked with Alan Wilson and Ned Johnson. So spanning sort of biochemistry and MBZ with a lot of input from Monty. His career's just taken off. He spent time as a postdoc at Florida State University of Florida, and then was curator of genetic resources and professor at University of Washington, and now he's curator of ornithology and professor at Harvard. So he's, I guess, the new Ernst Mayr. So you just have to live as long as Ernst and never be wrong. You'll do just right. So Scott has made many contributions, both in avian systematics and biogeography. More recently, he's had a really avid interest in promoting use of multilocus approaches in phylogeography and systematics, and that's what he's telling us about today, multilocus approaches to phylogeography and phylogeny in the Australian bird fauna. Oh, I might even recognise some species. Thank you, Scott, and welcome back.
Thank you very much, Craig. And I want to thank everyone for coming, and just really wonderful to see lots of old friends and meet lots of new faces as well. As Craig said, this journey really began for me right here, and it's been a lot of fun to look back and also to look forward as well. Now, I was told that I had to show an obligatory slide of me as a graduate student here. That's, I guess, one of the hoops you have to go through as these centennial speakers, and so here we go for that. This is me in 1986 in New Guinea. Just as a background, to let you know how I got started on this Australian journey, as an undergrad, I applied to work as a volunteer for Steve Pruitt-Jones, whom many of you know, and he spoke at the recent centennial meetings. He's still working in Australia as well. Anyway, we were in New Guinea for about 4 months, and it was just a dream come true for me. It's a paradise for ornithologists. And when that stint ended, I naturally was wondering, I remember vividly talking with Jim Patton and others here saying, I don't want to take classes my first year. I want to be in the field. And of course, Jim was all in favor of that. And so I managed to, I basically, in those days, applying for permits on the fly was easy. We've been complaining all morning about how difficult it is these days. Anyway, within a few weeks, I put together a number of permits to do some collecting in Australia. And that's really where I got started. Now, this shows me, as a young buck, if athletic prowess is any indication of my promise in the future, then I'm kind of sunk. This is me in recent years. It's just not going anywhere. Actually, I have practice, and I can keep it up now for a reasonable amount of time. But and now, of course, as Craig mentioned, it's been an absolute delight to head up the bird department at the MCZ, and I invite any of you to come and let us help you out. It's a fantastic collection, and of course not as much in the 21st century bioinformatically as the MBZ collection is. Of course, this is world-renowned as the leader in this area. But it's been fun to help bring it into the 21st century, and we've been working with Carla and others to computerize the collection and really make it available to people. There's a lot of, it's got a lot of diversity in the collection, many extinct species, and for example, one project that we've done recently involved looking at DNA diversity in ivory-billed woodpeckers, which, as all of you have heard, were reported in 2005, although it's unclear whether the report was genuine or whether they're really still with us. And anyway, there's a lot of opportunity, I think, for looking into the past with these old specimens. The majority of the specimens in the MCZ are actually from the late 19th century, so it's a really remarkable view into time. And this is our dubious distinction of the largest collection of ivory-billed woodpeckers, but it's one that many biologists have recently and the media has been quite interested in. And of course, we're all looking forward to next year, which is, of course, Darwin's birthday, and also, by coincidence, really, the 150th anniversary of the MCZ, and we hope to see you there. Now, my recent connection with Darwin involved visiting his house down it's about 15 miles south of London, and if any of you ever have a chance to visit, I would highly recommend it. It for many years was a school that was populated by school children and was really becoming quite run down in the 70s. And now they've refurbished it, and it's really a fantastic destination to visit. You can look at his desk and see his copy of Das Kapital, Karl Marx right there on the shelf. And of course, I went there to imbibe the true spirit of Darwin, not just to come as a tourist, but to really commune with our patron saint. And so this is a picture of me taking the walk around the sand walk, as Darwin used to do after writing every morning for a few hours. So I'll begin by with what better thing to begin with, really, than what I did here for my dissertation. And just a note to graduate students, you should never be ashamed of talking about your dissertation work, even decades after it's done. I mean, the bottom line is, most people in the audience, haven't read your papers. And even if they were on your thesis committee, they probably haven't read your thesis either. Now, I have actual proof that my thesis committee read my thesis because I have their signatures. But and I think that's good proof. But anyway, it's always the case that what you think is old is going to be new for many folks. So anyway, here's my stab at it, and this is work done in both the MVZ and in Alan Wilson's lab. It's delightful to see Alan Prager here. At the time, of course, Alan Wilson was a professor in biochemistry, and it was there that the polymerase chain reaction was first applied to many different species. And a little bit of trivia for you. What bird species was the first species to which the PCR was applied? Well, I had the lucky opportunity to put that on the PCR map. My hope is to be elected to the PCR Hall of Fame one day. But it was actually these babblers, and in some earlier work, looking first at cytochrome B, and then in this larger study at the control region. And I start with this slide to reiterate just the power of this molecule as a tool for looking at population history. You can see the different clades realized in this gene tree, for example, up here in Cape York in New Guinea, followed by this clade around the central east coast, and down on the southeast corner, all nicely registered as monophyletic clades in the gene tree. And then on the west side, we've got this large and widespread clade up at the top end, followed by a cluster of lineages in the Pilbara, the extreme western Australia. And so it's clear that mitochondrial DNA is hugely useful for mapping out the basic attributes of biodiversity. And the challenge that I and many others have been dealing with recently is, well, why should we look at additional genes? If we're getting such a nice picture of biodiversity, is there a need to look at additional loci to flesh out these biogeographic histories? And it's a recurring question because, just recently, just this year, a few weeks ago, I don't think it's even out in print yet, but two graduates of the MVZ, Bob Zink and George Barraclough, who was a postdoc with Ned Johnson, had a little review article in Molecular Ecology called Mitochondrial DNA Under Siege. And they had the feeling that mitochondrial DNA was being, its utility was being questioned. And I think it's a useful review to look at, but don't read it too closely. I think it's instructive in a lot of ways, and I think it really helps to. I think we need to be able to articulate why it is we need to look at so many genes in our studies, or if in fact we need to. And I think some of the answer to that question will come from, as usual, studies on model species, humans and Drosophila. And I think for phylogeographers of our ilk, it's come most recently in a greater realization about really the distinction between what gene trees say and what population histories say. I think at one level, we would all agree that the fundamental unit of interest among us is population history, species history. I mean, at some level, we could care less what the genes are saying because we're interested in organisms and their history and the vagaries of the environment as it's influenced that history. And so the challenge is to articulate what in fact we can say with a single locus versus what we can say with multiple genes. And so this is an example of a recent paper by a colleague of mine, Stefan Bench, at Lund University in Sweden, which I think illustrates some of these challenges. And so Stefan looked at a bunch of different markers in a group of birds called Phylloscopus warblers. They're old world warblers, small-bodied passerines. Trevor Price has studied them quite extensively. And very interesting data set. He looked at mitochondrial DNA. He looked at the W chromosome, one of the sex chromosomes of birds. He looked at the Z chromosome and several autosomes. And of course, as you can see, every gene tree was different. And the last thing that anyone would want to do with this data is to concatenate these genes into a single matrix and then analyze them in that way in an ensemble. And in fact, like other papers of this type, Stefan basically left it at this. In other words, without many methods really to combine these data other than concatenation, pretty much tried to weave a story around the separate stories told by each of these gene fragments, despite his clear awareness that all of these fragments are essentially telling the same story. Okay? So the challenge is to bring out that one history, that one story, from what appears to be several multifarious and often conflicting gene histories. So what I'll do today is tell you a little bit about some of our work trying to grapple with this heterogeneity at the level of gene trees. And we'll talk about a couple different, we'll introduce a couple examples from the Australian birds, looking at situations where phylogeny clearly is not an appropriate model for the particular scenario. Rather, a scenario involving gene flow or some sort of reticulation is probably more important and more appropriate. And then we'll end by talking about a couple examples where divergent history probably is a good description of the history and how we've tried to bring some tools to estimate that history. And one theme I'll have throughout the talk is that I think one of the big challenges right now is not so much estimating parameters. Once we've decided on a model, say we wanted to measure gene flow or measure phylogeny or population size, I think the challenge isn't so much estimating those parameters. The challenge really is figuring out what the appropriate model is. And only recently, I would say, even in leading systems like humans, have people done the sort of large-scale simulations that really allow us to decide, are we going to treat this as a diverging situation, as a situation involving gene flow, or population expansion, or whatever. So it's a challenging situation. Here's just a couple of the models we'll talk about. I'm sure most of you are familiar with all of these. And all of these it's an exciting time because many of these models are couched in the same types of population genetic parameters across different models. And so, for example, here is a diagram summarizing Peter Beerley's migrate package, which I'm sure many of you are familiar with. Estimates population sizes and genetic diversities, and also gene flow between populations. This model assumes that the migration pattern has been the same throughout the entire species history, which is unlikely to be the case for most species. Here's a model that has been developed by Rasmus Nielsen and Jody Haig, and it's the isolation migration model. It involves a divergence event right here, whose time you can estimate, and then it involves population sizes, both of the ancestral and the descendant populations. And on top of that, allows you to measure gene flow between the descendant populations. So it very opens up a lot of new possibilities. Now this model, I think, is still probably confined to 2 descendant populations, and therefore is somewhat restricted in its applicability. Now, a model that I've been involved with recently, which was originally developed by Liang Liu and Dennis Pearl at Ohio State University, we're calling a pure isolation model. And essentially by this we mean phylogeny. And this is one in which we essentially have successive divergence events at specific points in time, ancestral populations with their associated population sizes here. And what's exciting is that all of these models use as their currency, if you will, gene trees. But none of them are actually estimating gene trees per se. Rather, they're using gene trees and their uncertainty to get at a global estimate of the species history. And this, I think, is, we would all agree, really the modern focus of phylogeography. So Australian birds are a great system, and I once asked Ned Johnson whether Alden Miller would have minded if I worked on such a far-flung system as Australian birds, and Ned said no, he wouldn't have minded. He was very broad-minded in that way, and so I was able to proceed without guilt. Here's some of the systems we've been working on. These tree creepers, for example, have an east-west divergence across this region here called the Gulf of Carpentaria. And Australia, as students of Craig will know, has a very chock-a-block pattern of species ranges, wherein one can virtually predict the ranges of other species from constituent species with smaller ranges. And so, for example, the gray crowned babblers I did my dissertation on have a range virtually identical to that of the sum of these tree creepers. Here's some finches here, which I'll also mention, with an east-west range across the Gulf of Carpentaria. Again, matching very closely a range of the redback wren, a fairy wren, that my student June Lee is working on now, and I'll tell you a little bit about that as well. And so Australia is a great place to look at these questions because it hasn't undergone extensive glaciation. The ranges of species no doubt have changed dramatically over the last 15 million years, but have done so gradually without the rapid oscillations that have taken place in northern hemisphere continents. And you can see here an area cladogram developed by Joel Craycraft, in which he's mapped out the really predictions for phylogenetic diversity across the periphery of the Australian continent. And you can see here that this barrier B is the Carpentarian barrier, and that's predicted to be the primary split of species distributed around the outside of Australia. And on the east and the west, we have secondary, smaller breaks, which further increase the phylogenetic diversity. And so these are very useful predictions as we proceed in our work. This slide I simply put up to remind us that although I've tried to portray Australia as a gradually changing environment, it nonetheless has had lots of oscillations in its past, particularly with regard to sea level connections with other island systems in Southeast Asia. There was a lake, Carpentaria, in the middle of the Gulf now, which has been well described, and there's been lots of environmental history going back a few 100,000 years, where they trace the oscillations of the sea level. And it's also just a reminder that in the cases I'll be telling you about, for a variety of reasons, we have no compelling reason to worry that the biodiversity of New Guinea is intermingled with that in Australia. Although other systems that we're hoping to study soon are certainly would include New Guinea as a vital component. Just a travel log to show you. For those of you who've been to Australia, it's a great place to work. And there's not really much rhyme or reason to our treks over the years, except to say that as my family group has gotten larger, our expeditions have gotten shorter. But we have made a number of recent trips, and most recently by postdoc Chris Balakrishnan studying zebra finches and June Lee studying redback wrens. And I'm sure Craig will agree that the Australian hospitality can't be beat. In fact, those of you who know me, I've shown this slide at least 5 times in this room. So forgive me if you've seen this slide before, but it's a great place to work. Now, we've been focusing on a class of markers that we call anonymous markers. And, you know, as I give this talk, I'm telling myself that really, as you're all aware, we're undergoing a sea change of technology, and this is probably the last group of studies that we'll do as a lab in which we use these types of approaches using traditional dideoxy sequencing. Everything is changing. But what we've done is to basically clone out small fragments of anonymous DNA, and by that we mean DNA that doesn't register a strong blast hit when we blast it to the databases. We like sequence data for a number of reasons illustrated here, as compared, for example, with microsats. I think both markers have their strengths. We like sequence data because we can compare it with exactly the same statistics to mitochondrial DNA. We don't have to make any translation of scales. The mutational homoplasia is going to be minimal, and if we choose to do so, we can at least attempt to make gene trees, since they're a very useful heuristic tool. It's a lot easier to test for selection on DNA sequences, I would say, than on microsatellites. And so for a number of reasons, we've sort of converged on these markers. This just shows our protocol, which I'd be happy to explain to you later. It's not complicated at all, basically involving making a small plasmid library and picking loci essentially at random without regard to their variation, so we can get an unbiased view of the variation of the genome. Here's our pipeline for analysis, and again, I suspect things are just going to change dramatically in the next few years. Because we're amplifying diploid products, we have this problem of phase. I'm sure you've all run into it. Anyway, we deal with resolving the haplotypes computationally. Basically, we need to figure out what the actual alleles are we're dealing with. And we also have discovered in these studies a really high degree of indel variation. Many times indels occur as heterozygous, in heterozygous condition, making reading of the chromatograms challenging. But we've once we basically are comfortable with the fact that we might have non-recombining segments of DNA, we can then move on to our demographic inference. I think there will be a number of places in which the new technology will change this protocol. For example, 454 data will presumably make this issue of haplotype estimation moot, because that data is essentially looking at single molecule sequences with the idea that you're looking directly at a single allele as opposed to amplification and sequencing in which you're sequencing 2 alleles at once. Okay, so I think some of this will change, but it's also the case that, and I'd love to have a discussion with folks. It's unclear to me whether gene trees will remain a source of information, or whether we'll be moving to more elementary data such as SNP variation for most of our surveys. The combination will always be a challenge, and in fact, none of the programs we typically use incorporate recombination as a variable, and so there's always going to be challenges there. Okay, I'll just skip over this next slide since this involves the haplotypes, and rather move on to the birds. Here's the tree creepers, which you can see have a distribution very similar to the babblers, east and west of this Carpentierian divide. Rather than 2 subspecies, we've got 2 distinct species. And there's our sampling done over the last 10 years. Most of those specimens are in the Burke Museum. And here's our just a quick look at some of the gene trees we discovered. A couple things to look at here. The red here is the eastern lineages, and the brown tree creeper, and the green are the blacktail. None of the gene trees are what we call reciprocally monophyletic, right? There's no pure green and pure red clusters, so there's all intermingling among every locus. And obviously there's a huge amount of heterogeneity. And so we've adopted a a model summarized by migrate, essentially, in which we estimate gene flow within each of these two lineages. And I'll mention briefly some studies looking at gene flow across this potential region of contact. When we look at the population patterns, remember migrate can give us estimates of population size. We find quite a diversity of population sizes. Some of the noteworthy aspects are this population up here, which is fairly small and isolated, and we found a fairly low level of diversity. And this population here at Fitzroy Crossing, which we suspect is actually sort of a hybrid population, perhaps between other populations in the top end and this isolated Pilbara population, sort of creating a lot of diversity there. And the gene flow that we've looked at, just to summarize over many different analyses, essentially finds quite a lot of gene flow among these southern and central eastern populations, fairly restricted gene flow to the population in Cape York. Again, a fairly continuum across the top end with quite a lot of isolation by the Pilbara population. Now, one of the advantages to multilocus data, and in this data set we have about 15 genes in total, is that we're able to look at locus-specific effects. And so this analysis shows essentially the locus-specific estimates of gene flow as using this map over here as a key for these population designations. And a couple things to notice. One is that for any one locus, our confidence and accuracy of gene flows is fairly low. Each of these represents 95% confidence limits. The sum of the signal from many loci is quite narrow. The confidence limits are quite narrow, and those are indicated by these colored bars on the side of each graph. So we're definitely seeing an accumulation of signal with multiple loci. Another thing to notice is that there's across this region of potential contact between the eastern and western forms, which is indicated here in red, we actually see relatively low, we don't see strong evidence for gene flow between those two forms, albeit for one or two loci. And who knows, maybe those loci are under some kind of selection in an alternate genome. Maybe they're able to flow across this region easier than other loci. We don't really know. The point really is to show how looking at many genes at the same time can give you some interesting insights into outliers, etc. Now, to move on to the second system, the red-backed wrens, this is work done by graduate student June Lee, and he's one of these guys who I showed him a picture of one of these birds from a field guide, and he ran out of the office without ever asking a question. The next thing I know, he's off to Australia and sequencing the whole genome. He's got the focus necessary to get the job done. Anyway, he's been looking at red-backed wrens across this region, and these guys are interesting because traditional taxonomy studies had suggested that this population up here in the northeast actually should be more aligned to this top-end population rather than to the population in the southeast. So this would contradict Craycraft's area cladogram, which would have predicted continuity of this eastern set of populations. And so it would require a shift from the area cladogram proposed by Craycraft. Anyway, June cloned about 30 anonymous loci. Well, first starting with mitochondrial DNA, of course. This is the pattern that June discovered. Again, here's our three main sectors of the species. And what you can see is a reasonably clear picture of mitochondrial diversity. Here we've got approximate sister relationship between this Cape York and southeastern population. So this would contradict that taxonomy I just told you about. But at the same time, we see some, the story isn't completely clean. In other words, we've got some lineages from other populations persisting as close allelic relatives of this southeastern population, and reciprocally in this top-end population. So there's not evidence for reciprocal monophyly in the mitochondrial DNA, and so I think it's of interest to look and see what the nuclear genes are telling us. Well, again, if we sort of take a naive view of the gene trees, our eyes will gloss over it. We'll probably go home in a emotional funk because there's so much heterogeneity. And my advice, although I show these gene trees, but it's against my better judgment. My advice is just bypass the gene trees, go for the big picture, and I'll show you how we did that in this case. What you can see, of course, here is that we have quite a lot of variation. And we've looked at a number of different statistics, including Monty's S statistic, which measures essentially the degree of exclusivity between different geographic regions. And we found the whole gamut from gene trees which show quite a bit of structure, such as this one down here. You can see there's basically distinct monophyletic groups, to really the more common pattern where everything's a single big cluster. In terms of diversity, June found a lot of variation. This shows the average nucleotide diversity for the anonymous loci. Here's nucleotide diversity for 5 introns that we've looked at. Intriguingly, we find quite a bit more diversity in the anonymous loci than in the introns. We also find quite a bit more diversity than in the mitochondrial DNA, although the caveat, of course, is this is a protein-coding mitochondrial gene, ND2, so perhaps it's not a fair comparison. But I think it's clear that the anonymous portion of the genome has a lot of variation to work with. And in fact, I think it would be very important and I haven't seen a study which essentially asks, do we typically see more nuclear diversity than mitochondrial diversity within species? I mean, the data sets are out there, but I haven't seen a sort of a survey trying to ask whether this sort of question that folks have predicted for a number of decades actually has been borne out in the data. Here's an analysis using Structure, which is a program which allows you to demarcate populations based on the assumption of random mating within populations. And what this shows is that really what we're seeing in this complex is 2 major groups, as indicated by this diagram down here, which shows each column here is an individual bird, and you can see a major group consisting of Western individuals, and a second group which is essentially a fairly homogeneous group consisting of the 2 Eastern populations. And so in summary, this analysis shows that we're seeing a different picture than what was predicted by taxonomy. And I think the resolution that we've achieved with multiple nuclear genes is certainly complementary to, if not more so than the resolution we would have looking just at mitochondrial DNA. What I haven't shown you here, but what I'll show you in one of the next case studies, is how June's estimates of divergence time and population size, really, again, their uncertainty shrinks dramatically as he adds more and more loci. So we're seeing more confidence as we add more genes to the story. The next scenario involves these exciting birds. These are zebra finches. Now, zebra finches don't correspond to the typical trans-Carpenterian pattern that I've been showing you. Rather, they correspond to an equally common pattern in Australian birds, which involve species inhabiting the interior of the continent. And Leo Joseph in Australia has looked at a number of species with mitochondrial DNA, suggesting that there's often very little structure within this vast region of the interior of Australia. And so Chris Balakrishnan has been taking advantage of the zebrafinch genome, which is being sequenced, to study not only nuclear diversity, but to get an idea about whether the physical location of loci in the genome might be able to tell us something about the population history of the species. So he's developed sets of primers that he calls locus trios, which are essentially trios of loci with of known distance apart on the chromosomes. Here's 2 loci that are 2 kb apart, and here's a third locus that's 10 kb for the first one, 8 kb from the second one. And we chose these distances, which are pretty small on the scale of the whole genome, because what little we know about recombination in birds suggests that recombination is pretty high. And if we were to find, we would basically be surprised to find a correlation in the gene histories between loci even this close apart, 10 kb apart. Now in humans, it would be straightforward to find quite a lot of concordance between loci that distance apart. In birds, we suspect that recombination is high enough that we're effectively seeing genetic independence at this very short physical distance. And we'll talk a little bit more about that in just a second. The obligatory gene tree slide, again, lots of chaos. Here, this is FST. I didn't mention before. We're also looking at a bunch of wild-caught individuals from the Lesser Sunda Islands, which Ernst Mayr has a fascinating paper about the colonization of Australia through Timor. And zebra finches appear to be a species which, in fact, has colonized the Lesser Sundas from Australia as opposed to across Asia into Australia. So the FSTs that you see here are FSTs between the Lesser Sundas and Australia. And you can see there's quite a bit of variation. But again, no examples of gene tree reciprocal monophyly. Everything's showing mixed signals. In terms of overall genetic diversity, Chris found quite a lot of variation. Most of the loci he looked at were autosomal. And of those, 13 loci showed no variation within the Timor population. These are these loci in the gray boxes. They showed no variation within the island population, although in some cases substantial variation within the Australian mainland population. We also were able to look at genes on the Z chromosome, and intriguingly, these loci show really very low level of variation. You can see the level of variation in the Z chromosome genes are a fraction of the level in the autosomal genes. Now, the autosomal variation is substantial. I mean, this is basically 10 times the level found in the entire human species. So this is a big number in population genetics. This number is suspiciously small. We predict that Z chromosome should show about 3/4 the diversity in a typical autosomal gene. Why? Because for every 4 copies of an autosomal gene, there are 3 copies of a Z chromosome gene. However, we find substantially less than that variation in these Z chromosome markers, and so we suspect that selection may be driving these loci down to lower levels of diversity. This is our structure analysis, again using the 7 sets of loci, about 25 loci in total. Not so interesting, I suppose, the Australian population, the Timor population. But intriguingly, everyone's obsessed with this idea of diagnosability and monophyly. These populations, I can tell you right now, are actually not monophyletic for mitochondrial DNA. And yet, we're able to diagnose them in the same way that, many systematists would like to. So I think it's important not to conflate this idea of monophyly with diagnosability. I think if you want to diagnose something, well, you should throw more loci at it, and trust me, you'll be able to diagnose it in the most infinitely minutiae detail. This shows an analysis using Rasmus's and Jody's structure program. IM program. And in this case, what we're asking is, what was the fraction of the Australian population, or really, what was the fraction of the ancestral zebrafinch population that went on to found the Timor population, the island population? And that fraction is basically registered in this orange curve here. And basically, our maximum likelihood estimate is 0, a tiny fraction. You can see this curve essentially increases monotonically. And what this is telling us is that, as judged by these loci, Timor was founded by a very small number of individuals. If the model that IM employs, namely a single ancestral population followed by 2 descendant populations, is true, then we've got a window into the dynamics of that speciation, of that subspeciation. And so to end the zebrafinch section, I just want to talk briefly about linkage disequilibrium, since, I think this is going to be a really important tool in phylogeography. And in human studies, you're seeing the use of disequilibrium to understand population history increasingly. And so what Chris did is essentially measure disequilibrium among these locus trios. And just to show you how to read these graphs, essentially cells along the edge of this triangle document disequilibrium within these loci. And of course, disequilibrium, of course, means the correlation of patterns of variation at different sites, at pairs of sites within and between these loci. So when the correlation is very high, when the histories are essentially very similar, we register that as a black cell. When the histories are essentially uncorrelated, we can measure that as a gray or a white cell. And when we look at the Australian population, you can see that there's very little linkage disequilibrium going on. This is looked at one of our locus trios. Here are the variable SNPs within that trio. You can see that there's no strong pattern of linkage disequilibrium in this system, perhaps suggesting that this population has been large and freely recombining for a long time. Okay? Now, when we look at the same and other locus trios in the Timor population, we see a dramatically different picture. Here are the various locus trios from the Timor population. Actually, this is within a single locus. Here we have within a single locus here, within a single locus here, within a single locus here, clearly evidence for disequilibrium, at least among sites within a locus, within a few hundred base pairs of each other. Intriguingly, however, very little signal at larger distances among different loci. And so what this suggests to us is that something, perhaps a bottleneck, caused an increase in disequilibrium, but that disequilibrium is already beginning to decay rapidly. And that's evidenced by the fact that there's just virtually no disequilibrium at the scale of 2 and 10 kb. And we're hoping to use some of models by Monty and his students to estimate some population parameters from this kind of information. But we'd like to resurrect. Craig said I'm taking Ernst Mayer's job. That's absolutely not true. But it is true that I feel this. I think Mayer's founder event speciation has gotten a bad rap in recent years, and I think that we need to do a better job of asking, well, if it wasn't founder facilitated, what was the contribution of genetic drift to the scenario? And what we've done here is basically to sort of soften Mayer's founder effect term to founder facilitated, because we have ways to estimate the contribution of genetic drift to these divergence events. And we very much, and so our question really is, to what extent has this founder event contributed to the phenotypic and genetic divergence of these two subspecies? Now, let's see, I think the slide may be a little out of order, but let me just, there's a slide which I'll show you later. I'm sorry I don't have it here, but basically we estimated the contribution. What we did is we estimated a fraction. Oops, sorry, let me just go back here. Oops. We estimated the fraction of phenotypic divergence between these two subspecies. They differ in body weight, bill color, the breast band, amount of striping on the face. Using heritability estimates from various populations, we estimated the contribution of genetic drift to the phenotypic divergence of these various characters. And we found that between 20 and in some cases 90% of the phenotypic divergence was compatible with a simple model of multivariate divergence using the population sizes that we've estimated. So there's no doubt that selection has been important in this divergence, but to rule out any contribution of drift, I think, is probably a little short-sighted. Okay, I want to end by talking about a final model of divergence, which we can essentially call phylogeny. And when I met Liang Yu and Dennis Pearl at this workshop, actually, that Craig put together in Ohio a few years ago now, I was very, very excited. I think many of you know I've been interested in the question of species trees and gene trees for a long time. And Liang had developed a tool, essentially, for estimating the species trees directly. And this diagram is simply to review what most of you probably know already, and that is simply that gene trees and species trees are different. They're not the same thing, and that they differ in some of their quantities. So, for example, I think most of you know that gene trees can have a different topology than the species tree, right? I think one of these gene trees has a different topology, probably the green one. Gene trees have a different divergence time than species divergence time. And so, for example, whereas the gene divergence time for this green gene is way down here, the species divergence is way up here. And so, depending on the population scenario, those two quantities can be very different. And of course, species trees have as a parameter the population size, as sort of the volume of these diverging lineages. And with the new approaches developed by Liang and Pearl, we can now estimate not only our confidence in the species tree, but some of these other parameters, such as species divergence times and population sizes. The sort of interest in species trees goes back a long way. It's not a new phenomenon. One could argue that folks like Luca Cavalli-Sforza, working on human genetics, was basically interested in species trees. And I think it's just a curious, sort of consequence of our use of DNA sequences that we've sort of fallen into a local optimum, I would say. In other words, it's obvious that it's straightforward that I analyze DNA sequences phylogenetically. And I think to some extent, this may have prevented us from moving forward to that essential second step involving inference of a species tree from the gene trees. And that second inference is illustrated on this slide here, where most phylogeny packages essentially focus on this likelihood equation, which is the likelihood that most of us are familiar with, namely the likelihood of our data, our sequence data, given a gene tree. Okay? That's the normal likelihood that most of us are using. The second likelihood that Liang and Pearl unified with this first one involves the likelihood of a set of gene trees given a species tree. Okay? Renala and Yang, I think some of the work may have been done in Monty's lab, worked out this likelihood, and now we're able to put those two together essentially to estimate the species tree directly. This is Renala and Yang's equation. I won't bother to belabor it, but I think the approach used by Liang and Pearl is really exciting. My main contribution was to develop the acronym for this new method, which most of you know, I suspect. But it's, again, without going into any details, the challenge, I think, was to recognize that both gene trees and species trees are interdependent on each other. And so it's sort of like a circle, and you're trying to figure out where to jump in. How do you infer the species tree if you need gene trees for that, which themselves are dependent on the species tree? And again, without going into too much detail, it developed a really clever way of essentially starting the process off using a constrained prior on the species tree, and then going on to estimate the gene trees, and then from there estimating the species trees. Okay, I'm going to, in the interest of time, I'm just going to move forward a couple slides to show you this last system involving these grass finches. And this is work done by Brian Jennings, who's now assistant professor at Humboldt State University. These are in the same cluster of finches that the zebra finch is part of, and appear to conform to Craycraft's typical trans-Carpentierian pattern with two related forms in the west and a single one on the east. Now, Brian cloned a bunch of anonymous loci and made gene trees out of all of these. And his results are here. And what you can see is that the most common gene tree that we discovered had the expected pattern that Craycraft would have predicted, namely two western lineages as sister, the A and the H, and the eastern lineages, the Cincta, as the outgroup. And of course, all these are rooted actually with the zebrafinch sequence. But of course, we found additional patterns as well. And in about equal measure, we found a pattern wherein the Hecki subspecies in the west was the outgroup, and a pattern in which the Acutacauda subspecies of the west was an outgroup. And so, none of these, at some level, none of these trees are in conflict with each other. They're all having been generated by a single process, which it's our challenge to uncover. And so we applied the best method to this scenario, and here we have a very, very simple situation, just one allele per species. Nonetheless, that allows us to make some inferences about ancestral population sizes, which you can see here. We can discuss the confidence not in the gene tree, but in the species tree, okay? And so our confidence in this particular internal branch is 0.88, or whatever that's worth. And then we can also talk about species divergence times. And again, we chose this model pretty much because it seemed like the data was consistent with it, and one of the big challenges is to figure out whether that inference is correct. In Brian's study, he found a clear signal for increasing certainty with adding more loci. In this case, these are estimates of divergence time in these two top panels. In this case, this was really the only parameter. This is the ancestral size of the two sister species, the two western Acutacauda and Hecki species. Not much additional information added with more loci. But in general, we're getting a stronger signal with more patterns, with more loci. And in terms of the actual sequence data, it's very clear that a model in which the gene trees are allowed to vary, albeit in a correlated fashion, is a much better description of the sequence data than is concatenation, which would essentially involve forcing the same topology on all the loci. And these are just the likelihood graphs for the grass finches and another data set that we analyzed, use data with 106 genes, where this correlated model of a species tree is a very good explanation of data. Done some simulations. I won't bother to go through them in detail. I'll just end by pointing out that throughout these examples, we've seen a lot of lack of, you could say, in terms of the gene trees, we've seen a lot of lack of diagnosability. Lots of heterogeneity in the gene trees, very few examples of reciprocal monophyly. And yet we've been able to diagnose clades when appropriate. Additionally, it's intriguing that the phenotypes of these species are, I'm sure there are loci in the genome which are in fact fixed between these species, despite the fact that all these neutral markers that we've been looking at are clearly not fixed. And this, I think, shows a basic distinction, and I would say a target for future research. What are the loci driving the phenotypes apart, and how can we measure their dynamics on the scale provided by the neutral part of the genome? And so I'll just end by just mentioning a few conclusions that I think it's incumbent on us to. I think the era of gene tree perspectives was brought on by mitochondrial studies and has been extremely useful. And I think the challenge now is to get over the gene trees and focus on really what we would all agree is the more relevant parameter, namely the population history. And I think hopefully some of these examples have illustrated our attempts to do that. Don't worry about conflicts among gene trees. I'd say don't worry about gene trees at all, really. Just throw them into your favorite software and run with it. Discordance among gene trees doesn't mean a conflict of some sort. It could be just a fallout of the demographic process of a particular clade. And I've shown you how adding multiple genes can improve our resolution of particular phylogenetic problems. And again, a pitch to the theoreticians. What we really need now are ways of deciding which model we're going to apply. Are we going to assume a phylogenetic model, a gene flow model, whatever? That, at least to me, seems like one of the more challenging aspects of our work. Really, what's the initial step in deciding that model? So yeah, I'd like to thank all these people, and of course, my mentors here at the MDZ for starting me on this wonderful path. Thank you very much.
Okay, some folks may need to, including me, may need to get to classes. I invite the rest of you to please engage Scott with trenchant criticism, or advice, or friendly questions.
Yes.
How much do you think the age of divergence here makes a difference? Like zebra finches are pretty old Australian species. I don't know how old they're split with Timor.
Right. So the question was, how much do you think the age of the divergence of these species makes a difference? I think it probably makes a big difference in terms of the gene tree patterns we're seeing. Of course, it's always going to be the age as scaled by the population size. And in the case of the zebra finches, it's interesting in terms of just taxonomy. They are somewhat isolated. There is a closely related sister species, which occasionally gets placed in the same genus or different genus, but it's phenotypically very different patterns, and genetically, at least judged by mitochondrial DNA, appears to be fairly divergent as well. One way of sort of getting at your question is, again, getting back to the value of nuclear markers. In some ways, I would say nuclear markers are going to give us a longer window of time in which we can talk about the history. Mitochondrial DNA would coalesce fairly rapidly, and you can't do much with that single branch leading to your sister species. Whereas nuclear genes, although described by Zink and Barraclough as sort of lagging indicators of divergence, I would argue, well, in fact, we get a longer swath of time over which we can make inferences. And so, in the case of the zebra finches, though, my guess is that they're fairly recent. The divergence time between the Timor and the Australian population, I think we estimated as it was on the order. Well, it was on the order of about 1 million years, which actually isn't all that recent. So it's fairly old.
So my question, and sort of follow up, is that it's not just discordance among individual gene trees, but for individual gene trees, mostly rates, are you going to be able to pull out this information by bringing them together?
Are mostly, rate
rates,
differences?
Yeah, rates are called polytomies.
Right, yeah.
Polytomies, how are you, okay.
Right, yeah, that's a good question. Again, my advice would be not to obsess over the details of the gene trees, because it's almost certainly going to be the case that the tree for any one nuclear gene is not going to be well resolved. And I guess my question to you would be, why would you care if a particular gene tree wasn't well resolved? We're trying to ask questions about the species history, and I guess the comfort I would say is that even though that specific gene tree isn't well resolved, it's nonetheless adding some information to the overall picture of population history. Of course, all these programs, as you appreciate, I'm sure, essentially factor out the gene trees. In other words, they sort of try to integrate over the various possibilities for gene trees. So they're not necessarily dependent on having a single well-resolved gene tree. But of course, you're absolutely right. That if you have, the stronger your individual loci, it's likely that your population history will also be that much stronger.
So I have a bunch of different questions. The first one is, I'm really curious about your choice to continue with anonymous loci versus introns, given the many introns we have now.
Right. Yeah.
Is it worth the effort, do you think?
Yeah, that's a great.
Constructing the whole library.
Yeah, that's a great question. Rory's referring to there's been some recent work done from Sweden where they basically laid out, almost 200 intron markers for birds. It's very easy now to basically take primers off the shelf and just apply them to your species. Good question, and I think, yeah, we absolutely would rethink that. I mean, I would say, you know, it's not a huge amount of work anyway to make these loci, but I think there is a lot of merit to having perhaps even a standard set of loci that people look at. Absolutely. So, you know, I think, and you know, I think the jury is still out as to whether these anonymous loci are in fact more variable or not. In some ways, I'm not sure it's that relevant. There's going to be, the scale of information is going to be so much bigger. So who knows? We may well use it.
And then, with respect to best, am I correct in that it assumes that there's no gene flow
That's right.
among the types?
That's right. Yeah. Yeah, which is, you know, every model has its constraint. And one thing which we're working on now is basically methods to ask whether or not all of the loci in your data set are consistent with a model in which there's just divergence. In other words, of course, we don't know what the history's been, and yet we want to know whether the variation among loci is such that a single dichotomous history might not explain it well. And so, yeah, we'd want to flag those situations where gene flow is taking place.
One more. How does your population size estimates from best compare with, for example,